Hello everyone, welcome to Get My Class. So let's solve this question. It says the foreign DNA is inserted into the fragment that serves ampicillin resistant in PBR 322. So which of the following observation is true? Option A, non-recombinants will grow on ampicillin containing media and tetracycline containing media. Option B, non-recombinants will not grow on tetracycline containing media. Option C, non-recombinants will not grow on ampicillin containing media. Option C says non-recombinants will grow on ampicillin containing media, but not on tetracycline containing media. So let us try to understand the question. The question says the foreign DNA is inserted into the fragment that serves as ampicillin resistance in PBR 322. So which of the following observations is true? So first of all, what is PBR 322? PBR 322. So PBR 322 is a type of a plasmid. Okay? It is a type of a plasmid which usually are used as vector actually to transfer the foreign DNA. So we are talking about the plasmid which is naturally present within certain strains of bacteria. That is PBR 322. Now this PBR 322, as I told you, that it has restriction sites which will cleave and which will get cleaved, and it will help in insertion of the foreign DNA. So this can carry foreign DNA. Okay, this can carry foreign DNA. Okay, so it has its own site where it can carry foreign DNA. But purposely in the question, what are they saying? That, suppose this is PBR 322, they are saying that the foreign DNA has been inserted in the AMP resistant gene in PBR 322. So what is AMP resistant? That is ampicillin resistant. So here you have AMP resistant gene, that is, and this side, because PBR 322 has two, antibiotic resistant gene. One is AMP and the other one is TET resistant gene. Okay, so these are what? These are selectable markers. Okay, selectable markers. Okay. So these will actually help in checking out whether the organism will grow in the presence of ampicillin and tetracycline or not. So the organism which is where PBR 322 is there, of course they have resistance towards both ampicillin and tetracycline. Hence these will naturally they can grow in the presence of ampicillin and in the presence of what? In the presence of ampicillin and tetracycline. Why? Because they have resistance towards both. They have ampicillin resistant gene and tetracycline gene. But according to the question, they are saying that they are actually inserting the foreign DNA. So this is, suppose this is the foreign DNA and this is, okay, foreign DNA, and just put it as fDNA. They are inserting within the ampicillin resistant gene. So when we are inserting within the ampicillin resistant gene, what is happening? It will get inactivated. Ampicillin resistant gene will get inactivated. Why? Because it has been now altered, right? Because of the entry of the foreign DNA. So it will get inactivated, and this is called as insertional inactivation. Okay. So what will happen? The AMP resistant gene will get inactivated, and hence now the recombinant, wherever the foreign DNA is present, like the PBR 322, in where foreign DNA is present within the AMP resistant gene, what will happen? It will not be resistant to ampicillin. So it will cause loss of the ampicillin resistance. So it will not be resistant to what? It will not be resistant to ampicillin. Okay, resistant to ampicillin. Okay, but it will be still resistant to tetracycline because that has not been altered. Only AMP resistant has been altered. Now let us see. It says non-recombinants will grow on ampicillin containing media and tetracycline containing. Non-recombinants means which is having the normal PBR 322. So of course, yes, normal PBR 322, they are non-recombinant. That means they don't have foreign DNA. They do have resistant gene for both, that is ampicillin and tetracycline. And this is correct. Option B says non-recombinants will not grow on tetracycline containing media. No, because it will grow because it has tetracycline resistant gene, right? Because it is non-recombinant, we are saying. And anyhow, we are not altering the tetracycline here. So this is not true. Then option C says non-recombinants will not grow on ampicillin containing media. So non-recombinant, we have not touched the resistant gene there. So there it will grow on both, that is ampicillin as well as tetracycline. So it is ruled out. Option D says non-recombinants will grow on ampicillin containing media, but not on tetracycline containing media. So non-recombinants, they should be able to grow on both, because in non-recombinants, that means they don't have the foreign DNA. Please remember that. So they will be having intact AMPAR and intact TET-R. So they will be able to grow on both. So this also ruled out. So what is the correct answer? The correct answer is option A. So let us conclude the answer. The answer is option A. I hope you understood the explanation. Thank you, everyone.